Hey everyone, my name is Oswald Avery, and I'm going to be presenting my science fair project on what is the transformation principle along with my group mates. Do y'all want to introduce yourself? Hey, I'm Mason McCarty. And I'm Colin McLeod. I'm going to be going over the purpose of our experiment. Frederick Griffith discovered the transforming principle, which refers to how a substance can be transferred from non-living cells to living cells, causing the living cell to show characteristics of the non-living cells. We want to discover what exactly the transforming principle is. We want to know what part of the heat-killed S strain bacteria is responsible for the transformation of the R strain bacteria, which is making the R strain bacteria become virulent. Me and my team are going to be repeating Griffith's experiment to identify the component of the cell that's causing the transformation. To learn how heat-killed S strain bacteria cause the transformation of live R strain bacteria, please watch Griffith's Science Fair experiment video. Our hypothesis was that either RNA or DNA would be the transforming principle. I'm going to lead you through the experiment that we did. Streptococcus pneumonia has two strains. The first one, the smooth strain, is virulent, and then the second strain, the R strain, is not. For our experiment, we took live cells from a heat-killed S strain, and then we treated each with different enzymes. And then we treated this with the R strain. We incubated this and spread this into petri dishes. In the first batch, we destroyed the polysaccharide coat of the bacteria. In the second, we destroyed the lipid content. In the third, we destroyed the RNA. In the fourth, we destroyed the proteins. And in the fifth, we destroyed the DNA. So I actually have a graphic that illustrates this. Okay, so in this one, as you can see, we have the polysaccharide degrading enzyme, we have the dead live S strain bacteria cells, and then we have the R cells. Over here, we have one with RNAs. Then we have this one with trypsin. Then over here we have one with lipase. And then the last one has DNAs. So altogether, there are 5 different ones. My groupmate is going to go over our results. So overall, for our experiment, we wanted to see what would happen to the rough strains. If there was a smooth strain bacteria found in the petri dishes, it would mean that the rough strain was transformed. The only petri dish that failed to convert the rough strain into the smooth strain was the one that was treated with DNAs, which means that it contained no DNA. This was the result from our experiment. For our conclusion, we found that DNA is a genetic component that is being transformed, or that does the transferring. No DNA means no transformation. When DNA is destroyed, R strain bacteria cannot be transformed into living S strain bacteria. Therefore, DNA is responsible for transfer of genetic material. This means that our hypothesis was correct. I have a little graphic to show again. So in our graphic, you can see how the strains that were previously rough strain transformed and now show characteristics of the S strain. And you can see this in every single one, except for in the one that was treated with DNAs, because since the DNA was destroyed in this one, the DNA wasn't able to transfer the genetic component, and therefore the R strain was not able to change and become the S strain. Thank you for watching our presentation. We hope you liked it. Bye.